Hey, hello guys, welcome, vanakkam, vandanam, namaskaram. Today we are going to see molecular basis of inheritance part 2. Already we have seen part 1. If you haven't seen, just go and check it out, okay? So now we are going to start with packing of DNA helix. Okay, so first of all, we have to know what is this DNA helix. See, this particular concept is very complex in terms of reality, but in this book it's very easy. Okay, see, totally in our humans there is 2.2 meter long chromatin. Okay, if you just take out the genetic material in a particular cell and if you're just stretching out into the form of thread, then it will be about 2.2 meters in humans. Okay, and after that, coming to the number of base pairs, see, there are about 3.3 into 10 to the power of 9 base pairs in haploid condition, and in diploidic condition in humans it is 6.6 into 10 to the power of 9 base pairs. Okay, so now the distance between each base pair is going to be 0.34 nanometers, right? So when you multiply this 6.6 into 10 to the power of 9 into 0.34 nanometers, then it gives you 2.2 meters. Okay, so this is the length of all these DNA or all these chromatin or all these genetic materials. If you just stretch it out in the form of thread, then you will be getting this. Okay, so then after that, coming to this other organisms, see, for an example, the pi into 174 is having about 5,386 base pairs. Okay, wow. Then in bacteria page it is about 48,502, and also in E. coli it is about 4.6 into the power of 6 base pairs, right? So okay, in one alone will be chromatids. Here will be nucleotides. Here it is nucleotide. Okay, so since, see, just 2.2 meters is not a small length, right? It is 2.2 meters and it is being stringed. It is being compact. It's very complex. Okay, and then it is being stuffed into a single cell, and it's not a simple deal, right? So that is why we are going to check about this topic. See, this sort of arrangement is actually two different types. In eukaryotes it will be one type, and in prokaryotes it will be one type. So coming to eukaryotes, it is a cell. Okay, so where in a cell, since there don't be any nuclear membrane, so since there is no nuclear membrane, it does not mean that this, one second. Okay, so it does not mean that this DNA will be split into everywhere. Okay, it will not be just all around the cell. It will be in some parts of the cell. Okay, and then they will be all compact. Okay, they will be forming a loop. They will be forming a big loop because there will be some positive proteins, and then there will be this negative DNA. So since positive protein, and around this positive protein, this negative DNA molecule will be there, and then that will be forming a loop. That loop is known as nucleoid. Okay, this loop is known as nucleoid. Okay, this is in prokaryotes. Coming to eukaryotes, here we are going to check about a few things. First of all, in eukaryotes, you are going to check about positive proteins. See, we are having lysine and arginine as positive proteins. What determines this positive protein is that in the side chains they should be having amino acids. Okay, they should be having amino acids. The abundance of amino acid is the thing that actually checks whether they are positive or not, or it checks the magnitude of the positivity. Okay, so it's all based on amino acids. Okay, then coming to histone octamer. See, we are going to check about histone octamer. We are going to check about the DNA that is around this histone octamer, and then we are also going to check about what also this linker DNA, then their base pairs. Okay, all these things is solenoid, super solenoid, then nucleosome. Here only you are going to check about nucleosome. Here it is nucleoid. Okay, wow. Okay, see, now coming to histone octamer. See, histone octamer is a structure which has positive proteins in it. It has positive histone proteins in it, such as H2A, H2B, H2A, H2B, H3 and H4. Okay, all these will be 2, 2, 2, 2, 2. In a histone octamer there will be 2, 2, 2, 2, 2. Okay, and then they will be all arranged in a compact way, and surrounding them there will be this DNA. Okay, this DNA molecule will be around them and be just coiling this positive protein. Okay, ma'am. So if you are just taking out this DNA, okay, this DNA will be having around. Okay, So this DNA is there, right? So this is just a DNA that is around this histone octamer, or this histone octamer is also known as new body. Okay, so this will be having around this particular DNA that is around this new body or this octamer, right? That will be having around 200 base pairs. Okay, around 200 base pairs. Okay, so this particular one structure, okay, this total structure with this DNA and then with this histone octamer, also this DNA will be next going to, okay, this DNA will be next going to the next cell, okay, to the next around 200 base pairs. Okay, around 200 base pairs. So next, this DNA will be moving. This DNA will be moving. Okay, it will be moving, and then it will be going and checking about next another histone octamer, which will be also having these proteins. Okay, see, before moving, what will be happening is this DNA is there, right? So these two, so there will be an entering DNA. See, from this is how the DNA comes. Okay, and this is where the DNA leaves. Okay, so there should be a notch, there should be a tie. So that tie will be done by H1. Okay, like this only the tie will be. So that will be done by H1. Okay. Here there are totally 2 molecules. Here there are totally 2 molecules. Here totally there are 2 molecules. Here also there are totally 2 molecules. So totally there is 8 molecules. There are totally 8 molecules in this new body alone. Then along with that, if we are just taking out this total part, there are totally 9 molecules. Okay, along with this H1. Okay, so like this, it will be going through. So this DNA will be going to the next new body. Then like that will be going on. And when you are viewing it with a microscope, okay, just a normal microscope, a little bit more advanced than the normal microscope, it will be like this. Okay, so how is this structure? This structure is similar to the beads on a string. Okay, so it will be similar to beads on a string. Yeah, so these are those important concepts. So since this new body, okay, or this histone octamer is positive in nature, this DNA is actually negative in nature. That is why it will be coiling. Okay, so this DNA is there, right? The DNA which links this new body and this new body, right? So this DNA will be having around 60 base pairs. Okay, 60 base pairs. So yeah, so these are those important concepts. Okay, so now we are going to check about the order. Okay, we are going to check about the order. So first the DNA. So when this DNA is with this histone octamer, or also known as new body, okay, with this histone octamer, DNA plus histone octamer, which will be giving us what, this nucleoid. Okay, so this particular structure, this total, when this histone octamer and this new and this DNA joins, will be giving us nucleoid. Okay, this nucleoid. Nucleoid, and then later on this nucleoid will be forming as solenoid. Okay, this nucleoid will be forming as beads on a structure. Okay, beads on a string. Beads on a string like structure. Okay, so then this will be forming as solenoid. What is the spelling? I'm very weak in spelling. Solenoid. Okay, then this will be forming as super solenoid. Okay, super solenoid. Okay, then later this will be, okay, then later this will be forming as chromatin. then chromatid, then chromosomes. Okay, then only it will be forming as chromosomes. Okay, so this DNA and this chromatin, then it will be forming as chromosomes. Okay, so then if you're just checking it out, later see, the beads on string structure in chromatin is packed to form chromatin fibers. Okay, so that are further coiled and condensed at metaphase stage of cell division to form chromosomes. Yeah, so this chromatin fibers, okay, chromatids are also known as chromatin fibers. Which are being condensed at metaphase. Condensed, okay, at metaphase. Okay, to form chromosomes. Very important. Metaphase, prophase, or these all are very important where questions can be asked, I think. Okay, I'm not a teacher, I'm just a student. So the packing of chromatin at higher level requires additional set of proteins that are collectively referred as non-histone proteins. Okay, non-histone chromosomal proteins in a typical nucleus region of chromatin are loosely packed. Okay. So if you're just seeing out a chromosome, okay, so this is a chromosome. So in this, there will be some parts where it will be loosely packed. Okay, so this loosely packed region is there, right? So that is actually known as loosely packed. So that is known as euchromatin. Okay, so euchromatin is actually transcriptally active, loosely packed. Okay, so this is known as euchromatin. Okay, so this is actually transcriptally active. Then later, see, this one is here, right? So this one next it will be, it doesn't mean like the top chromatids will be loosely packed and down chromatids will be tightly packed. It does not mean like that. I'm just writing it. Okay, so here it will be tightly packed. Tightly packed. So when it is tightly packed, what does it mean? It is actually known as heterochromatin. Okay. Okay, and it is inactive. Okay, and then this is only light. Okay, light strains will be there. Here it will be both. Okay, both light will be there and also both dark will be also there. Okay, so it's like a spider web. Okay, then from here easiness starts. Okay, just this one is actually there. So yeah, here actually it will be stains will be dark. It is not, sorry, they did not give it both tightly and so wait, light. Here it is not both. Here only dark stain is there. Okay, only dark chain is there. Okay, so now coming to the next concept, which will be the search for the genetic material. Okay, so coming to the search of genetic material, first of all, we will be checking out a few terms. See, in the year of 1926, okay, in the year of 1926, okay, there was a quest to find the location. Okay, there was a quest to find the mechanism of genes. Okay, there was this quest to find the mechanism of genes, and then a few scientists, okay, naming like Gregor Mendel, Walter Sutton, Thomas Hunt Morgan. Okay, all these persons are there, right? Who are Gregor Mendel is there, then Walter Sutton is there, then at last R. Morgan is there. So these people are there, right? So they all have narrowed the location of chromosomes. Okay, they have just said that chromosomes are there. There is a genetic material. Okay, there is a genetic material. Okay, and also they have narrowed the research of chromosomal locations. Okay, they have narrowed in sense, they have shortened the location. That means they have made a way where other scientists can use that way to find the chromosomal locations. Okay, so here, if you just come to here, the search for genetic material. People, scientists have got it out. So since our Mendel has said that there is a factor, that factor is the thing that is responsible for the inheritance, and also other people have started to find some comments. Okay, some other people have started to prove that it is correct. So what scientists have done is they have started to see what is there. Okay, what is the thing that is actually there? What causes this genetics? Okay, what is being inheritance? How it will be there? How it is being formed? How it is being replicated? How are we gaining it? Okay, so all these things had started. Okay, so first in the very beginning, if you just check it out, the first research. Okay, so the first research was done by Mendel only in the year of 1865 itself. He did. Okay, what did he do? He found about the inheritance by using pea plants. Then after 4 years, it was done by Frederick Meister. Okay, he is a person who found nuclein. Even though these two timelines, see, 1869 and 1865 isn't that much different, right? They both are literally parallel, but our Mendel did not come to know that there is something like this nuclein. Okay, maybe if he had known about this nuclein, maybe he would have said that factor is none other than nuclein. Maybe he would have, but since he did not know what is it, so he did not make any such sorts of assumptions. Okay, then after that, in the year of 1928, see, 1869 is the last year in the 17th century. Then coming to the 18th century, we are in the 21st century. In the 19th century, the last research was held at 1869 only. Then coming to the 20th century, in the year of 1928, okay, Friedrich, here it is Friedrich, here it is Friedrich, here it is Friedrich. Okay, here it is Friedrich, here it is Friedrich. Okay, so Frederick Griffin in the year of 1928. So he did a research based on this genetic material, where that is what we are going to see now. Okay, based on this genetic material, where he used Streptococcus pneumonia. Okay, Streptococcus pneumonia, and he just used mice. Okay, he used mice. Okay, so yeah, what does a griffin mean? Griffin is a creature. It is a majestic imaginary creature, right? So yeah, this actually Griffin symbolizes loyalty, Griffin symbolizes bravery, Griffin symbolizes independency, Griffin symbolizes ethics. Okay, so coming to here, the search for genetic material. So in the first, who did it? It was done by Frederick Griffin. Mostly last name only will be asked, so that is why I said about Griffin. Okay, so coming to Frederick Griffin, in the year of what? 1928. See, here I have written all the years. Wait, not here. Here in this page, and then in this page, I have written all these years. We are going to see that later. So, Frederick Griffin, in the year of 1928, why is they come up? In the year of 1928, he did a research. Okay, so what he did is, okay, so he had taken a few things. First is he has taken Streptococcus pneumonia. Okay, fortunately or thankfully he didn't use humans. See, now we are going to see about Streptococcus pneumonia. Streptococcus pneumonia. See, it is actually of two different types. Okay, yes, actually it is smooth. See, this Streptococcus pneumonia can form smooth colonies, smooth, shiny colonies, and then it can form rough colonies. Okay, see, this smooth and shiny colonies are there, right? So these organisms are having in their outer surface, and this is what the cell is. Okay, so they can form a shady membrane, which is actually mucus. Because in their membrane there will be glycocalyx. See, calyx. Glycocalyx. Spelling is correct, huh? Okay, wow, it's correct. Okay, so where this glycocalyx will be forming mucus. Okay, which causes death. Okay, which causes death. Okay, but whereas this is unharmful. Okay, this is unharmful. Okay, ma. Okay, so first there are a few continuous done. Okay, a few continuous experiments was done, and in this process, a lot of rats had gone away. Okay, so continue, Ranjali. Okay, so coming back to the concept. See, first what he did is he had taken a mice. Okay, and then he had with this mice he had injected this R. Okay, we'll be saying it as R strain and S strain. We will be saying. Okay, this is known as S strain, and this is known as R strain. Okay, so he had injected R strain. Okay, and fortunately the mouse actually lived. Okay, then next mice or mouse? Mice only. Okay, so this mice he had also injected S strain. Okay, and now it had died. Okay, then later what he did is he had also injected this mice. Okay, along with this mice he had actually injected heat killed bacteria. Heat killed bacteria. Heat killed bacteria. Okay, and now the mice lived. Fortunately, okay, the mice actually lived. When he normally itself heat killed in sense he produced heat and he killed that bacteria, and then he just injected it to this mice, and then now here in this heat killed bacteria, in this heat killed bacteria, it is a virus, na? Okay, it is a bacteria only. Sorry, thank God, it is a bacteria only. It is not a virus. Okay, so heat killed the bacteria. So where now the mice actually lived. Now what he did is he had actually injected mice, and along with this mice what he did is he had taken heat killed bacteria, and also he had added R strain bacteria. Okay, R strain alive. Okay, this bacteria is alive. He did not kill it. Okay, sorry, bacteria is not, every time I cannot write bacteria, lazy. Okay, so then later what happens is, see, from this there will be the genetic material. This genetic material is fortunately it is alive. Okay, so the genetic material which is in the cell of this heat killed bacteria had got transformed to this S strain bacteria. Okay, then later, since it has got that particular DNA or that genetic material, now this bacteria had started to develop that mucus. Okay, so that because of that mice died. Oh my God, mice died. Unfortunately, the mice had passed away. Okay, now you can say that R strain bacteria had got transformed into S strain bacteria because the genetic material from the S strain bacteria had got transformed to the R strain bacteria. Okay. This is what they have done. Next is, we are going to check about biochemical characteristic of transformation principle. I like biotechnology, but I don't know anything about biotechnology. Biochemical, these are biochemical with the spelling, characterization of a transforming principle. Characterization of transforming principle. Okay. See, where here, if you're just checking it out, who are the scientists who are involved with this? Next step. See, in the year of 1933 to in the year of 1944. Okay, here only this S and R. Okay, so see, in the year of 1933 to 1944, okay, 11 years, okay, 11 years, okay, they have done a research, and from that research, what they concluded is that they have concluded that DNA is the genetic material, okay, DNA is the genetic material, and also in this process also, they have killed a lot of rats, hamsters. I have a hamster in my home, and I have to protect it with, okay, I have to protect it with from scientists. Very hard job. Okay, so these are those scientists. Who are they? Oswald Overy is there, and oh, these names are headaches. Okay, Oswald Overy are there. Colin McHugh is there. Then Macklin McCarthy is there. Okay, McDonald's only is coming to my mind. Nothing is coming. Okay, so Oswald Overy, Colin McHugh, and McCarthy. That's even enough. Okay, Mikhail McCarthy. Okay, so see. They have considered a lot of vessels. Okay, so about how many na? Five. They have considered 5 vessels, and in these 5 vessels, what they did is they have included genetic materials. So, yes, heat killed the bacteria. Yes, heat killed. Yes, heat killed. Same bacteria only. Okay, yes, heat killed bacteria only they have taken. Okay, here also yes, heat killed bacteria. Yes, heat killed bacteria, and here also yes, heat killed the bacteria. Okay, so now what they did is and also along with this yes, heat killed the bacteria. They have also added R bacteria. Okay, R strain bacteria, R strain bacteria, R strain bacteria, R strain bacteria. Okay, here also they have also added R strain bacteria. And also they have started to add some other enzymes. Okay, laces enzymes, not lace, eating lace. It's very bad for health, they say it, but what to do? It's very tasty. Okay, so along with this, but I don't like to eat it for some reason, I don't know. Okay, see here also with this they have started to add some other enzymes. For example, they have added here carbohydrates. Okay, protease, then lipase, then RNAs, then DNAs. Okay, so these are those enzymes that they have also added with this. Okay, and now they have seen, and now what they did is they have taken the bacteria. Now they have injected this to the mice. Okay, and at last it results here the mice died. See, because the carbohydrates means from this, okay, from this every carbohydrate that is with this S killed bacteria and with this R killed bacteria will be died. Okay, that will be taken off. Carbohydrate cannot be died. It will be taken off. Okay, it will be destroyed. You can say here protein will be destroyed. Here lipases will be destroyed. Here RNAs will be destroyed. Okay, and here DNAs will be destroyed. From now even you can conclude that here mice died. Here also mice died. Here also mice died. Here also mice died. And there was this one and only one lucky mice which lived. I think they are in their hell. That they have killed these many mouse. Okay. Okay. Now, next is they have, see here the genetic material is DNA that they have concluded now. Now, this protein, this enzymes are here, right? This wasn't that much acceptable, okay, because of some reasons. Some scientists had denied that, no, I won't accept it, okay. So what some other people, now DNA is the genetic material that they have concluded, but even though still some more research had taken place in order to confirm it 100%, okay, because we want some sort of confirmation with these sorts of researches, okay, because these are life-changing, these are human total life-changing researches, right? So if in case they were able to confirm now, so still further researches can be taken out, right? So for that purpose, this one wasn't just like that accepted. Still further some other scientists had researched and then only they have accepted. So here comes our great, here the space won't be enough, so we will save it and then we will okay. Now okay. Now here if you are just checking it out, we are going to check about the next topic, which is the genetic material is DNA. How can you conclude that the genetic material is DNA? So coming back, oh, that PDF is there. I cannot open and I don't have that much of what calmness. So I will just write the name and in the year. Okay, Alfred Hershey and Martha Chase. Alfred Hershey, Martha Chase. Okay, so what did they do? This research had taken place in the year of 1952. Okay, the same year only, in the year of 1952 only, X-ray diffraction data had also, that particular X-ray diffraction, that research has also taken place, which is in the beginning of this lesson itself, and it was done by Wilkins. Okay, I think it is Wilkins only. So it was done by Franklin and Wilkins. Okay, so Myris Franklin and then Rosalind Franklin. Myris Wilkins and Rosalind Franklin. Myris Wilkins and Rosalind Franklin. Myris. Here we will write. Myris Wilkins. Wilkins. Rosalind Franklin. These names are really such a headache. Okay, so Myris Wilkins and Rosalind Franklin, in the year of 1952, they found X-ray diffraction. Okay, why two scientists? Isn't one scientist enough for finding something? Okay, so what here they did is they have again taken some vessels. Okay, okay, so they have taken E. coli. Okay, here also it is fully of E. coli. Once again. Okay, so since he was shouting, I've taken my best friend. Okay, so with this E. coli, what he did is he had taken also this bacteria page. The terrifying look, no bitings. No. Okay, so he has also taken this bacteria page. Okay, so in one bacteria page, he had given sulfur. Okay, so sulfur 35, okay, which is radioactive. Okay, then in another bacteria page, he had given what? He had given phosphate 32, which is also said as radioactive element. Okay, so what is, see, you have to know one thing, that is in the capsid. Okay, in this capsid, so this is in the capsid. Okay, in this capsid there are a few elements such as sulfur, hydrogen, oxygen, nitrogen, nitrogen and carbon. Okay, so in this he had added, so since into this capsid what he did is he had added sulfur. Okay, sulfur 35. Okay, so and also there is this inside genetic material. Here is also inside genetic material. See, how cute. This is why I like nature very much. Okay, so and also here is also this genetic material, and inside this genetic material we know that there will be phosphodiester bond, right? So there will be phosphodiester bond, right? So to that phosphodiester bond, in this he had substituted it with the phosphate 32, which is actually radioactive element. Okay, so where he had given, okay, where now what he did is, since here there will be E. coli, and here also there will be E. coli, right? Okay, so this particular mechanism is actually that it will be giving its genetic material. It will be giving its genetic material to this E. coli. Here also be giving its genetic material to its E. coli. I'm very slow because my pet he can be a little bit more aggressive when I'm very fast. And that too, now there is a lot of change in the room for YouTube, and also in my dressing style also I have changed it with lungi. And he's very not familiar because he bites my leg when it's visible. Okay, when it's with pant he bites, when it is with lungi he cannot, right? So that is why, and I too don't like, it's very hot in here. Okay, so I know I'm very hot, but the temperature I'm talking about. Okay, so now it will be passing its genetic material to this E. coli bacteria. Okay, now what they did is they have taken this capsid. Okay, so now it would have passed it to here, right? Okay, so they have taken just the E. coli and then they have centrifugated it. Okay, so first what will be happening now? First it would have passed its genetic material to E. coli, and that passing is there, right? That is actually known as infection. Sorry, injection. Infection. Okay, then blending. Okay, blending. Then after that, he centrifugated them. Okay, so now the genetic material is there. Okay, so E. coli will be there, and along with this E. coli, Also they did this blending. Okay, now later what they did is they centrifugated it. Now if you just check it out, there will be two different parts. There will be two different parts, where in the top part and then the bottom part. Okay, so it will be drawn in two different colors. So top part is actually known as superent. No bitings. Okay, so it is known as superent, and then the bottom part is actually known as pellet. Okay, superent and then pellet. Okay, so when he did the check, okay, when he did the check for sulfur, okay, he cannot find any sulfur over here, because S35 is in the capsid venti. S35 is in the capsid venti, and the capsid did not form. Okay, did not transform any. It did not come here. Okay, but when he did for here, okay, when he did for here, phosphate will be in what color? I don't know in what color will be phosphate. Okay. So here, no bitings, I said, Papa. He's sleeping. Okay, I wish I was a bird. That I wear a bird. Okay. Here, if you just check it out, here also you did the same blending, and again when it was coming here, okay, when you take it out, when you are taking it out, and also he bites very good. It's paining, no bitings. Okay, so superent, and then pellet. Okay, again when it was centrifugated, here he was able to see DNA will be always down only. Okay, DNA will be always down only, since it is actually kind of heavy. Okay, DNA, and in DNA he was actually able to find this phosphate 32. So since it was able to, since we can find it, you can say that DNA is the genetic material. Okay. So next, after this, coming to the properties of genetic materials. See, there are a few properties which has to be followed by a genetic material. If it is a genetic material, for example, if you are considered as a genetic material, okay, you should be having some ideal properties. One and only if those ideal properties are being matched, then only you will say that you are a genetic material. Okay, so what are the properties of genetic materials? I'm very slow now because I'm very tired of standing. Okay, see, RNA is here. RNA is here. This RNA would have got changed. Okay, see, in the very beginning, in the total life era itself, that means from the beginning of the life origin till now, in the very beginning, it was only virus. It was only a few creatures which had been formed, like prions, virions, then virus, like this only bacteria. Then from that only we people have evolved. Okay, ma'am. So in the very beginning, organisms, okay, the life organisms, the life origin organisms had only RNA. Then after mutation and after evolving only, this RNA had got transformed into DNA. No bitings. He doesn't bite with a force. He doesn't bite to attack me. He bites, and it wouldn't even pain if he does not have the sharpness of his beak. But since he's having, it's paining. Okay. Why am I saying all these stories? We have to discuss this one. Oh, you are distracting me. Okay. So coming to here, properties of genetic material. RNA has got transformed into DNA. Okay. See. Here it has got evolved. Coming to the evolution process, since it is not that much important in this topic, we'll be reading it then, but it's okay. We will now even we'll check it out a little bit. See, first was prion. Okay, next is virion. Okay, then later virus. Then later bacteria. Okay, then after bacteria, protista. So after protesta, yeah, like that only we have all got originated. Okay. Okay. So yeah, it all got transformed. Okay. See, in prions only RNA is here. Here also only RNA. Later this RNA. Okay. Later only this RNA. Then from here also it was only RNA. Then only it was no biting. Then DNA. Then DNA. Okay, later only it has formed into DNA. Here, if you are just checking it out, here we don't even know that whether it is living or it is non-living. Okay, because it can be crystallized. Okay, yeah. Okay, so coming to RNA versus DNA. What are we going to see? Oh my gosh, my leg is paining. And this lungi is not comfortable since it's the first time I'm wearing. Okay, so coming to here, RNA versus DNA. I think already you would have known all these. Like this versus alone. See, RNA is actually reactive, very reactive. Here it is less reactive, or it is stable. It is actually stable. Why so? Because see, here this RNA is having OH minus. Okay, sorry, it is having OH in its second carbon. Okay, here it is having only H in its second carbon. Okay, wow. This OH is here, right? Which is in the second carbon now. So this oxygen. Okay, here there is no oxygen. This oxygen is what? Very reactive. Okay, reactive, and then here it is no oxygen, so that means it is less reactive. Here it is highly mutatable. Here it is less mutatable. Okay, ma'am. Then after that, here it is having uracil. Here it is having RT9. Okay. Then other than that, here it is actually single-stranded. How can I write? There is no space. Okay, here it is single stranded. Here it is double stranded. Okay, so 1, 2, 3, and then 4. Okay, so I said here it is single, double, single. There is okay. I know I'm talking something, right? Because I'm very tired. Okay. Now we have said about some characters, right? We have said okay, so we'll complete with with this event. So I said that there are a few things, okay, there are a few properties that a genetic material should have, right? So those properties we are going to see now. Okay. So the first property, okay, better to be a very ideal or to be a very good, obedient, okay, the favorite genetic material, okay, like me, very obedient, okay. So we want a few things. First is that it should be, it can do its own replication. replication should be there. Okay, replication should be available. Then after that, it should be more stable. It should be highly stable, and it should be having slow evolving. Okay, evolving. Okay, evolution should be there, but the evolution has to be very slow. Okay, then after that, expression of characters should be there. Expression of characters in sense like how, see, first of all there will be DNA, then it should be replicating, then by transcription process it should be forming as RNA, then from RNA it should be translation, then from translation it should be proteins, and proteins will be forming enzymes, enzymes will be later showing its own properties, right? So like that. Okay, like that only it should be. Okay, so we will complete with the RNA world. No. Okay, so see, I'm using the word so a lot, right? So okay, all these. First genetic material was itself RNA. RNA was the first genetic material, was to be considered as the first genetic material. And later only that got evolved. Okay, so that is why being stable is good instead of being changing. See, DNA is now considered as genetic material and RNA is left out. Very poor RNA. Okay, see, now this RNA is actually responsible for all the metabolisms. How so? Here I am going to write the central dogma process, even though I have written it already once in the first part itself. It's okay, I am going to write it again, just check it out. Okay, so first the DNA will be there. Okay, now this DNA molecule, this DNA is there, right? This genetic material will be replicating, will be blue color. Replication. Then by transcription, RNA molecule will be formed. RNA molecule will be formed. Then later on, with this RNA molecule, okay, what it will be doing is it will be doing translation. Okay, and later on it will be forming DNA molecule. Sorry, proteins. It will be forming proteins. Where these proteins are those which are responsible for the what? For characteristics expression. Characteristics expression. Okay, so this is how the metabolisms are taking place. Next is also, see, protein synthesis is actually translation process. That is what I have said now. So translation is done. Okay, so translation process is done. Now this RNA itself is actually three different types: mRNA, rRNA, tRNA. Okay, like that it is itself is actually of three different types. And then see, from DNA, if it should be formed into RNA, then there are a few things that should be noticed. Okay, so first is splicing is there. Okay, splicing is there. Then heading is there. Then is capping. Okay, not heading. Splicing, like that. All these processes are there. That will be reading there. And at last, first in the very beginning will be HNRNA only. From DNA, HNRNA will be produced. That HNRNA will be later transformed into DNRNA. Okay, sorry, it will be transformed into mRNA. HNRNA, from HNRNA by some process it will be transformed into mRNA. Okay, and other RNAs are there, right? tRNA is there and rRNA is there, right? They will be transformed in a different way. That is not in our portion. Okay, so yeah, with this we will be completing this total video. So I hope you all liked it and it was helpful. So thank you. We'll be meeting in another amazing video. Bye bye.